Okay, histology we have already discussed about the histology of renal corpuscle, basic histology. That means the wall of this Bowman capsule is made up of a squamous epithelial layer, and the neck, from the neck portion onward, that tubule system in nephron is made up of cuboidal cells, and the cuboidal cell, in case of proximal convoluted tubule, they are with the brush border. Later on, that again, when they reach into the Henle's loop, it becomes again squamous epithelial. When the Henle's loop ends and they start this distal convoluted tubule, again the cuboidal cells start developing, and they remain in a cuboidal cell form, but they don't have any this brush border, microvilli like brush border structures. Okay, so that same thing is shown here again. But one more point they have highlighted here is a JG complex. So this is a ascending loop of Henle. The cell which are attached with the artery and this afferent arteriole and efferent arteriole, that portion of cuboidal cell epithelial cells in case of ascending loop of Henle now become modified. They are not normal epithelial cells, but they are now called cuboidal. They are not normal cuboidal epithelial cells, but they are now called as a macula densa. And this junction portion where these afferent arteriole and macula densa come together, that is a JG complex. Okay, here all these blood vessels internally line with this endothelial cells, and that is a capillary portion surrounded with the podocytes, and all that remaining gaps between the glomerulus are packed with the mesangial cells. Mesangial cells serve as a packing material in a glomerulus. Here again the same labeling, and we have seen previous slides, we have seen on this diagram. Here in nephron physiology, we don't have to go in detail how that absorption, reabsorption, secretion take place in the formation of a urine, ultrafiltration thing. But the machinery required or involved in this ultrafiltration nephron physiology is all that. If you start from here, it's a JG complex, that macula densa, then glomerulus, endothelial cell, podocytes, then mesangial cells, then lining in a Bowman capsule. There you will see that simple squamous epithelial cells, then neck portion, cuboidal epithelial cells with a lot of brush border thing. Here again squamous epithelial tissues, then again cuboidal epithelial tissues, and here it goes on with the cuboidal epithelial tissues, and slowly it should change into the squamous epithelial tissues. Histologically, it looks like this. Okay, now we'll go a little bit in detail, discuss detail about the juxtaglomerular apparatus. Now, here this is a juxtaglomerular apparatus. Distal convoluted tubules. We'll see some component first. This green color indicate the distal convoluted tubule. This is a afferent arteriole, and this is a afferent arteriole. Okay. Now here again we'll see some label macula densa portions. Then in centrally junction of all these three tubules there is a lacis cells, and here they have shown along with the endothelial cells, they shows one more type of cell is a juxtaglomerular cells. Now when these distal convoluted tubule come back towards the Bowman capsules and lie close to the vascular pole of a renal corpuscles between the afferent and efferent arteriole, they form a triad-like structure, junctions, that is called the JG junctions. Now, in this region, the muscle cells in a afferent arteriole, muscular cell layer in a afferent arterioles is modified. That muscle cells become large, and they shows a rounded spherical nuclei, and the cytoplasm of these specialized muscle cells in afferent arteriole, they contain granules that can be stained with a special method, so which you don't see in another arteriole. These cells are called as a juxtaglomeralular cells, and they are innervated by unmyelinated adrenergic nerve fibers. These juxtamedullary cells are regarded by some as highly modified myoepithelial cells as they contain contractile filament in their cytoplasm. Now, the wall of this distal convoluted tubule is also modified at the site of contact of arteriole. So here the cell lining is densely packed together and they form a columnar type of cells. In rest of the thing you will find they are in a cuboidal epithelial cells, but the area which is in close contact with the arteriole is rather than cuboidal, it has become a columnar, a tube-like structure cells, and these cells are called as a macula densa. This lies in close contact with the juxtaglomerular cells. They are in very close contact with these muscle cells, and they together then form the juxtaglomerular apparatus. So JG complex and macula densa, they together form this juxtaglomerular apparatus or a complex. Now, these JG juxtaglomerular cells, if you see under electron micrograph, you will find that they contain a crystalline structure, enzyme renin, which is a substance responsible for production of angiotensin 1 and angiotensin 2. Now these both angiotensin 1 and 2, they are related with the increasing blood pressure and they influence the adrenal cortex. So their role is in a maintenance of a blood pressure. So here in this flow chart you can see how JG complex maintain the blood pressure by producing initiating the conversion of angiotensin to angiotensin 2 and all that vasodilatation, vasoconstrictions, then aldosterone secretion, everything. But because of this our histology course, we don't go much in the physiology portions of this JG complex. You can remember that JG complex have a role in maintaining the blood pressure, increasing or decreasing the blood pressures, and how it is formed, what are the cell type. Now here in next few slides we will see how exactly it look like JG complex look like in a slide when we stain it. So here we'll try to see the components here. So proximal convoluted tubule, these are the proximal convoluted tubule. Then macula densa, and here these increased tubular structure cell indicate the macula densa because the rest are cuboidal and these are all columnar type. These are the renal corpuscle portion. This much is the renal corpuscle portions, and these are the distal convoluted tubules. Distal convoluted tubules, when come in near contact with the arterioles, it forms this macula densa portions. Okay. Here they have shown the P. P indicate the podocyte, nucleus of that podocytes. And E indicate the endothelial cells, P indicate the podocytes here, and A indicate the afferent arteriole. Now here in a color photograph of a slide you can see macula densa. Now clearly you are convinced that this cellular type in a distal convoluted tubule is a cuboidal and here it is a columnar. So we say that this is different cells and this is a macula densa. This just to revise the cellular type in a convoluted tubule system is here see we are saying many times that they are cuboidal cells. In case of proximal convoluted tubules, you will find there is a lot of microvilli and the basal membrane or basement membrane. Okay, so here it is a distal convoluted tubule. It is a cuboidal structure with less number of microvilli, and that you can differentiate in these two areas. This is a distal convoluted tubules. You don't see much microvilli here. It is a more microvilli and the cuboidal cells. So here it's a brush border structures in a proximal convoluted tubule. All these are the cuboidal epithelial cells. Distal convoluted tubule, you will not find any such brush border structures, so you can identify it as a distal convoluted tubule. Wherever there is a microvilli structure is present, you will see here a lot of mucus accumulated or sections. You can see these microvilli distinctly, which are absent in case of distal convoluted tubule. That spaces you can identify it as a proximal convoluted tubule. Okay, so vasa recta structure. Here, lot of blood vessels. You see a network of lot of blood vessels. That way, you can say that this is a vasa recta or that blood vessel portions around the loop of Henle. Okay, this collecting duct and thin segments and capillary electromicrograph here. So, lot of capillary are around the collecting tubules and thin segment of Henle and thick segment of Henle is there around. There is a lot of capillary portion is there. So all these are the capillaries around the Henle's loop here. These are the capillaries, and around this, these are the Henle's loop. These are the collecting tubules. In collecting tubule again, there is no brush border, so these are all columnar. Cuboidal epithelial there is there. So this is a cross sections here of the collecting tubules. You will find many such sections of the collecting tubule in a LS of kidney. So if you see such structure, this is what is a duct, collecting duct cut into the vertical sections. Again, this is a kidney structure, and here you will find that distal convoluted tubule, proximal convoluted tubule, collecting tubule, and everything. So more and more color photograph I am trying to show you to get you acquainted with the. When you see that slide under microscope, you can identify and remember this point. Again, this is a medulla portions of kidney, and so here this is the collecting duct. This is a Henle's loop. Okay, and this is a thin loops of Henle. Same thing here again, kidney in a medulla. So these are the cuboidal cells of the collecting duct, and these are the lumen of collecting duct. These are the all collecting ducts lumen and the cuboidal epithelial lining. So again the sections in a collecting tubule, sections of a collecting tubule showing the lot of cuboidal cells. Again here collecting tubules and cuboidal cells, but here in a loop of Henle, the thin squamous epithelial tissues you will see. So on this is a not a collecting tubule, it is a loop of Henle. So on this kidney topic in mammalian histology, frequently asked questions are these. So this is a give an account of gross structure of kidney. That way you can describe that all cortex, medulla portions, pyramids, and that hilus portions, then uh, that middle blood vessels and everything. Then explain microscopic structure of uriniferous tubule. Here we have to describe the structure of nephron. Explain histological structure of juxtaglomerular complex. You describe that glomerulus or renal corpuscle and describe about the cell type, juxtaglomerular cells and the macula densa cells and their functions and their structures. Then if they ask write short note of JG complex, you draw diagram and explain JG complex. Then case of medulla of kidney. In medullary portions, you have to draw the collecting ducts, loop of Henle, and majority of group of Henle, thin and thick segment you have to show, and the collecting tubule in a medulla portion. If it is a cortex, you have to show glomerulus, proximal convoluted tubule, and distal convoluted tubule rather than the collecting tubules, and the Henle's loop are absent in the cortex portions of TS of kidney. Then uriniferous tubule structures, that is the nephron. You have to describe the structure of renal corpuscle. Only the glomerular worm and capsule structure you have to describe in this. Then gross histology of kidney. Now here you have to describe all this above together. And that way, these four short questions are generally for 4 marks, and these long questions are around 8 marks. 6 to 8 marks.